Well, in the last video, we talked about how you turn genes on and off. All right, and you did that with something called methylation, all right, of the CPG island. Well, this island, all right, was out here in the front of your gene. In other words, here's your TATA. All right here. And up here was your C's and G's. You might have a few afterwards. All right, and these might get methylated to turn it off, or you leave it unmethylated to leave it on. Well, it just so happens that not only this was the on and off switch, not only do you control the on off right here in this region, in this pre-promoter region, all right, but you also control, okay, so the pre-promoter region, there are lots of names for this region, but the pre-promoter region can also control quantity or volume. All right, so it's not just on and off, but it's also volume. And by volume I mean how many RNAs you make. All right, so you're making like a bunch of them. You make 3 a second, or maybe you make 1 every 30 seconds, or you make 1 an hour, or you make 1,000,000 in 1 hour. All right, well, however many you make is also controlled by this region here. In other words, how efficient is that copier? In other words, did you tell the copier to run and make you 20 copies, or did you tell it to make 1 copy? All right, what's going on here with the copier, and where are the controls for that copier? Well, they're also in this region, and they're mixed in with these C's and G's. And the example I think I gave in class was something called GAGA. I'll just use it. It actually does several things, but we'll just say that this thing is an example of a transcription factor. Transcription factor. It's a factor. It's a protein that controls the rate of transcription. It controls the rate, like how many you make at any given time, of transcription. That means how many copies you make. And this can be a sequence, just like here we had C's and G's. It might be a sequence like this one, GAGA, G-A, G-A. So maybe let's see if we can fit G-A, G-A in here somewhere. Sorry about the writing. And just like RNA polymerase sticks here, there are going to be other proteins that are going to stick to the GAGA. And these proteins that stick to here either help the copier out or they hurt it. In other words, they either slow down the copier or they speed it up. So maybe GAGA, in this case, speeds up the copy machine, the RNA polymerase, which is sticking here. RNA polymerase sticks here, GAGA sticks here, and so that means go 10 times as fast as normal. If the GAGA doesn't stick there, then you make just 1. All right, maybe 1 an hour. Sticks here, you make 10. Maybe you make 100 or 1,000 an hour. And so this is the way you control the rate of basically the transcription. The gene needs to be turned on in the first place, so you can't be methylated, or that's going to turn everything off. All right, but if it's unmethylated, you might also have a transcription factor, a little sequence of letters in here in which a protein can stick, and that protein will help out the copier. It will actually make it go faster, or it actually could also make it go slower. And a lot of times these transcription factors can be influenced by the environment. So the environment can influence whether they stick or not. Now, it's hard. I don't have another name. So this is the GAGA protein, and this is the GAGA sequence. So the GAGA protein may not stick if it's a certain temperature or a certain pH or some other difference. And so this is a way for your body to kind of measure the environment, whatever's going on in your body. It might affect how well it sticks here. And if it sticks, it increases this. If it doesn't stick. So this means that maybe this sticks better when it's hot. So if your cell is getting a little warm, I'm just making that up. GAGA doesn't work that way, but other transcription factors do. So if it's warm outside, basically you get warm, your cells get warm, and so this sticks better. And when this sticks better, you make more of these. Maybe this is a protein, maybe this is a gene that produces something in your sweat. So maybe you need to make more of this when you're hot, and that would be how that is controlled. So now we have not just what is a gene, how do we turn them on and off, but we also have a way of controlling when we do it. So these transcription factors, all right, control when, all right? And not only that, they can be influenced by the environment. So they can adapt your chemistry, all right, to what's going on around you. So this is how your body can change. You go out, you get hot, you get cold, you get whatever. You don't necessarily turn genes on and off when those things happen. What you do is you control how much of various things are made. And this is how you adapt to the environment. All right. And so this is our lesson today. So we're going to need to know these things for the next test.